Today we have to continue with the drug interactions. Last session we have to discuss about the various types of drug interactions. First is site-wise drug interactions, and on the basis of actions like last sessions with the classification of drug interaction, either in the form of site-wise or internal and external, and on the basis of mechanisms, the drug interaction. First is pharmacodynamic and pharmacokinetic. These are the two mechanisms of drug-drug interactions. We have to discuss. Out of that, pharmacokinetic interactions, they are further classified 4 types on the basis of their ADME characters. Absorption interactions. During the absorption, what type of interactions was occur? Either enhancing or decreasing the dissolution as well as absorption rate of concerned object drug. होते rejoin. They continue on that. All of you heard my voice? आवाज येतोय? Let's start.
ब्रेक होता है सर थोड़ा
ब्रेक होता है, थोड़ा नेटचा प्रॉब्लेम आहे, इश्यू बघूया. डायरेक्ट आता शेअरिंग करतो. What are the slides I have prepared? The last session आपण absorption plus distribution बघितलेलं आहे. In the pharmacokinetic interactions, you have to see about the absorption interactions and distribution interactions. All of you seen my screen? Okay, the third part of the pharmacokinetic is then metabolism interactions. For the metabolisms, either metabolism is faster or slower, depending on the enzymes present in the metabolism interactions. Generally, these metabolisms, whatever the drug alterations of object drugs, we have to study in the metabolism interactions with the help of some enzymes, either enhancing the metabolism's rate as well as some enzymes inhibit the metabolism rate of object drugs. These metabolism interactions are further classified on the basis of enzyme actions. First, enzyme inductions. In these enzyme inductions, they increase the rate of metabolism of object drugs. Here, for metabolism of object drug, enzymes are helpful to increase the rate of that particular drugs, object drugs. And second, the mechanism of metabolism interaction is enzyme induction. Enzyme inhibition. Second is enzyme inhibitions. Here, in enzyme inhibition, name itself indicates the enzymes are inhibit the rate of metabolism of object drugs. This metabolism interaction is important one for the object drug to give the desirable action, desirable response of pharmacological drug. These are the two classes of metabolism interaction. First is enzyme inductions, second is enzyme inhibitions. Let's see about there some examples of enzyme induction as well as some enzyme inhibition actions. Here, object drugs, for example, in enzyme inductions, the rate of metabolism of concerned drug was increases due to the enzyme, and that reaction is called as enzyme induction interactions. Here, some example object drug is in corticosteroid, oral contraceptives, coumarin-containing derivatives, phenotines. These are the object drugs when react with the or they given with the barbiturates. They decrease the plasma level of concerned drugs, decrease the efficacy of object drugs. When they combine with the barbiturates, they decrease the concentration of object drug at plasma, as well as the efficacy of that particular drug was decreased due to the presence of this barbiturative enzyme induction interactions. Similarly, another example in enzyme induction category, oral contraceptives and oral hypoglycemic agents. These are the object drugs. When they given with the reform species, there is a decrease in the plasma levels, but increase the metabolic rate. Once the metabolic rate is increased, there is a reaction or efficacy of concerned drug was decreases. Second class of metabolism interaction is enzyme inhibitions. Here, enzyme with the help of enzymes, there is inhibition of the metabolic rate or decrease the metabolic rate of concerned drug. Decrease the metabolic process means increase the plasma level concentration indirect. For example, here in enzyme inhibition interactions, alcohol, they react or they given with the disulfurams, they increase in the plasma level in the form of acetaldehydes. So those are suffering from the alcohols majorly. For that conditions, disulfurams is the drugs was given to avoid the consumption of alcohols. The unwanted effect whenever you take the alcohols, there is an omitting. In presence of disulfurams, there is an increase the plasma level in the form of acetaldehyde. Alcohol is present, but in the form of acetaldehyde. Another example is tyramine-rich crude. You have to take us along with the MO inhibitors. They enhance the absorption of unmetabolized tyramines. Means there is a decrease in the metabolisms, may be harmful to the body. Another example in enzyme indication inhibitions, coumarin derivatives. When coumarin derivatives combined with the drugs metronidazoles or phenylbutazones, they increases the activity, anticoagulant activity. These are some examples of metabolism interactions, either enzyme inductions or enzyme inhibitions. Last class of pharmacokinetic interaction, that is the excretion interactions. All of you know, majority of the drugs are excreted through the kidney, near about 95%, and rest of the from sweat or other process they excrete from the body. Here, the object drug excretion pattern may be altered due to the excretion interactions. We have to study here the object drug pattern of excretion. Generally, the major mechanisms are involved in the excretion interactions are alteration in the renal blood flow. If occurs, there is alteration in the excretion of object drug. Second one is alteration of urine pH, maybe alkaline or acidic pH. They have further excretion of object drug. Competition for active secretions, again the major role during the excretion interactions. And lastly, forced diuresis. Sometimes it requires diuresis. These are factors, or these are the mechanisms involved to alter the excretion of concerned object drugs. These are some examples of excretion interactions. The major mechanism due to the change in the active tubular secretions. There is examples, penicillin or cephalosporin or dinidixic acids. If given along with probenecids, they elevated the plasma level of acidic drugs. There is an increase in the plasma level due to the presence of probenecids. Probenecids is a beneficial combination with increasing the concentrated plasma level with this penicillin and cephalosporin. It may be alter their excretions due to elevated plasma level of acid drugs due to आवाज येतोय change in urine pH screen दिसते change in urine pH. The example is amphetamines. They combine with the precipitant drugs, that is antacids or thiazides or acetazolamides. They increase the passive reabsorption of basic drugs, the effect of object drugs on object drug due to the presence of these precipitant drugs. If amphetamine is given along with the antacid and thiazides or acetazolamides, they increase the passive reabsorption of basic drugs and increase the risk of renal toxicity due to the change in the pH. Third mechanism of excretion interaction is a change in the renal blood flow. For example, lithium bicarbonate is the object drugs. When they given along with the NaCIDs, there is a decrease in renal clearance of the lithium. Is the major cause of renal toxicity. These are some examples related with the excretion interactions while changing in the urine pH, change in the renal blood flow, changes in the active tubular secretions. These are the pharmacokinetic interactions: absorption, distribution, metabolism, and excretion. These are some examples related with the parameters. Next mechanism-wise interaction is the pharmacodynamic interactions. Nothing but the how our body acts over the response of object drugs. Here, the activity of object drug at the site of action is altered by the precipitant. It may be direct or indirect interactions must occur during the pharmacodynamic interactions. These pharmacodynamics are interactions are classified on the basis of direct pharmacodynamic interactions or indirect pharmacodynamic interactions. If you see the examples in pharmacodynamics, some actions of drugs gives antagonisms, some gives addition, some gives potentials. On that basis, they are categorized pharmacodynamic interaction. First is antagonisms. All of you know, all of you heard antagonisms, nothing but it gives the opposite actions, opposing action of the object drugs. Here, interacting drugs having opposing actions. For example, acetylcholine and noradrenaline having opposite effect on heart rate. This is the best example of antagonisms. ACH and NA having opposite effect on heart rate, either enhancing or decreasing the heart rates. Second, pharmacodynamic interactions may be additions of actions of both the drug, object drug as well as precipitant drugs. The drugs having the similar actions, they give the sum of individual response of concerned drugs. There is simple summation 1 plus 1, that is 2. That type of action given in this category. For example, CNS depressant like sedative and hypnotics. Both the drugs give the CNS depression. That is additive actions. Another example you have to give in addition or submissions like sulfur methoxygen combination along with trimethoprim. This also gives the addition reaction to the antibacterial properties. Another drug is synergisms or potentiators. Here, it is the enhancement of actions of one drug by another. Example in this category, synergism or potentiator. Alcohol enhances analgesic activity of aspirin. If aspirin given with along with alcohols, the activity of analgesic of aspirin was enhanced due to the presence of alcohol. This effect is called as synergism effect. Due to another drug, potentiate the activity of one drug. There is enhancement of drug action of one drug by another drugs. The simple example is alcohol enhancing the analgesic activity or NSAID activity of aspirin. This is related with the pharmacodynamic interactions. Here we have to see consequences of drug interactions, what will happen during drug interactions. If major drug interaction is occurs, then is this type of interactions is life-threatening, maybe dead light situation occur due to the major drug interactions. In moderate conditions, deterioration of the patient status. If drug-drug interaction is moderate, the patient status hampered. Like current scenario, someone suffering from the coronavirus, then other people are seeing काय म्हणता येईल एखादा गुन्हा केल्यासारखंच म्हणजे त्याच्या स्टेटसचा डिक्रीज झाल्यासारखं कोरोना एखादा झाला आजूबाजूला कोरोना सापडला. In a minor drug interactions, if drug interaction minor case, there is some effect on our body and they neutralize within short times. These are the consequences if drug and drug actions was occur in major, moderate and minor conditions. This type of majority action was occur or observed during the drug-drug interactions. How will you reduce the drug-drug interactions? Firstly, identify the patient's risk factors. Means some patients are allergic to the concerned drugs, so avoid that particular drugs or identify the allergic drug of particular patient and avoid it. Take through the drug history, whatever the responses of drug to the concerned patient, you have to get the history from patients, whether this drug is beneficial to the particular drug, particular patient or not. You have to take their history. Be knowledge about the actions of the drug being used. Means whenever you are given the drug along with another drugs, all the mechanism actions of all the drugs should be known by the physicians. Due to known actions, maybe reduces the risk of drug interactions. Consider therapeutic alternatives. These are the another parameter for reducing the risk of drug interactions. Means replacement of safer drugs with the other drugs, alternative drugs. You have to find out. And avoid complex therapeutic regimen when possible. If possible, avoid the combination drugs during the therapy. And educate the patients regarding the drug-drug interactions, what are the drugs prescribed by the physicians, pharmacists. Role is important to educate concerned drugs before meal, after meal, like that. Whatever the actions was observed of concerned drugs, you have to educate or intimate to the patients. So, to avoid the risk of drug interactions. And finally, monitor the therapy, whatever the drugs given to the patients, particular physicians or pharmacists or nursing ward nurses will monitor the therapy of person drugs. What will happen with the particular patients? Either they are suffer with the effects of undesirable effect. If observed, immediately intimate to the physician and go with the alternative to minimize the risk of adverse effect of particular drug-drug interactions. These are the parameters you have to utilize for the reducing of risk of drug interactions. And last segment, it is the detection of ADRs, that is adverse drug reactions. How will you detect? All of you know, before the market, whatever the drugs, they are undergo the clinical trials. Nowadays, you have heard corona vaccines. They undergo in phase 2 trials, phase 3 trials, whatever the response is recorded, you have to, whatever the adverse reactions recorded. Here you have to see the pictures. Here one calculator to calculate the, this is required for the calculation, how much percentage of concerned drugs when enter into the market, what will happen with the particular response you have to calculate in the form of percentage. Nowadays, you have to observe in the bulletins, new channels, the death ratio of corona patients are in India hardly 2 to 3%. This, with the help of this calculations from area to area, you have to calculate how many percent. In Brazil, 10%, in US, 5%. These are the calculated data or responses to the concerned virus. Here, some physicians are involved in the studies, some pharmacists are involved in the studies of ADR detections. There are some different ADR techniques with the help of their survey, preclinical studies, clinical trials, after exposed to the certain area, after marketing, like coronavirus introduced firstly in Greece, they are exposure of that vaccines to the particular area. So unwanted, undesired effect, you have to noted and detected and noted and implemented according to that, whether it is continue or discontinue from the market. ADR detection, because no drug is absolutely free. They are having some side effects, some toxicity, because this is a foreign part of which is enter in our body. Maybe safe, maybe toxic, maybe gives undesirable effect when certain areas. For that, we have to detect the unpredictable adverse reactions and through various approaches are used as follows. I will detect through the various approaches. First is cohort study. Here, near about 500 patients we have to study over there in a cohort study to evaluate the outcome of the drug exposures. Means here you have to study near about 500 patient response, and with the help of that response, whatever the drug undergo exposure to the concerned therapy, for example, hydroxychloroquine, which is beneficial during the corona treatments. So, whatever this type of study we have to observe and noted as a cohort study. Near about 1 to 500 patients frequency you have to observe during the detection of ADRs after the exposure of drugs. Second is spontaneous report. Name itself indicates that spontaneous means continuously report of concerned drugs. When prescriber prescribe particular drugs and they observe the continuously during the investigations, nowadays some Mumbai's hospitals undergo a clinical trial with the Covishields. Indian corona vaccines. They spontaneously reported against that particular vaccines. And particular agency are giving the related informations and provided that information to the prescribers, and which is helpful for the ADR minimizations. These reports. Third detection approach is a review of vital statistic. For static, whenever the statistics occur, this computer is activated, this calculator is activated to know the statistic of concerned drugs. Unexpected increase of adverse reactions. This statistic is national as well as international review of the concerned drugs, which is help to identify of the unexpected increase of the reaction of concerned drugs. Because national and international, maybe environment are different. Some environmental conditions, particular drugs gives lesser adverse reaction. Some gives the higher adverse reaction. Depend on environment conditions. Also, responsible for the adverse reactions. That statistic we have to review in the detection of ADR, a review of vital statistics. And lastly, case control study. Case control study, nothing but nowadays corona vaccines everywhere. The announcement of corona vaccines will start within 50 days, 60 days. Whatever the study related with that particular vaccine, Covishield in India, whatever the cases observed, control study, with the help of that study, whatever the ADR was happens in the case study controls, case control study we have to observe. For particular patients, they undergo voluntarily for the treatment of concerned drugs before they enter into the large populations. Whatever the study, we have to report it as a case control study, and this reported study they undergo for the approval of new drugs. That type of study we have to observe. During these trials, nothing but in short, we have to call as trials before entering into the society. Either some part of the earth or some part of the nations, they undergo trials. Whatever the trials, whatever the response, we have to study. Concerned either beneficial, either adverse reaction or no reaction with the particular drugs. That study you have to report it as a case control study, which is helpful for the getting approval from the FDA, related FDA, either it is safer to use or not safer. This is important study as a case control study. In case control study, doctors, pharmacists, nurse, certain area physicians also involved in the case studies. These are the four methods to detect the ADRs, adverse drug reaction. Cohort study, it is related with the around the intervals of the 500 patients, and whatever the responses, you have to record it as a cohort study. Spontaneous reports means after entering into the market for limited populations, you have to report spontaneously regarding particular drugs, either beneficial or not beneficial. Vital statistic, it is related with the international and national responses toward the drug that will be reviewed and made the statistic. Like examples, coronavirus statistics, death ratio and recovery ratio are the different one. In India, recovery ratio nowadays 73% and death ratio only around 2%, whereas in other countries, recovery rate near about 50 to 60, or maybe more than our country, and debt ratio may be less or more with our country. Our country in corona debt ratio, they got the third place in the corona debt ratio, means less than 2%. Another one or two countries are involved, whereas in recovery rate, it is come under internationally in the 10th positions. Might be. The recovery rate in India is might be good one as compared to the other countries. So these are the different approaches where you have to detect the ADR before entering into the market. So this part of, as per your syllabus is concerned, ADR detections, type of drug interactions, type of adverse reactions. You have to study here. Predictable, unpredictables, classification of ADRs. Predictable, they are classified three. Unpredictable, again, they are three. Drug interactions on the basis of various, they are classified three types: site-wise, mechanism-wise, and other beneficial-wise. They are classified. Mechanism-wise, they again classify pharmacodynamic, pharmacokinetic interactions. Pharmacokinetic interaction again classified into four subtypes, and pharmacodynamics are two types: direct or indirect. And finally, detection of ADRs. Because this detection is important, because no single drug is a same drug. They are having some side effect, little bit side effect to our body. So to overcome that side effect, how to control? For that, this detection is important. So here up to today, the first unit of your pharmacy practice was over. In the next class, we have to start with the second next topics, next unit. Whatever the material available, according to that, we have to continue the next sessions. Like, for example, next probable topic is distribution of drugs in the hospitals. Most of the parameter we have to cover in the hospital pharmacy. They again and again come in the distribution of the drug in hospitals. Hardly within one or two lectures, we have to finish that part. Whatever the part we have to cover earlier, that part are fastly moved during the lectures. Like outpatient flow charts, inpatient flow chart in hospital. Already you have to study in hospital pharmacy. General, apart from that flow charts, we have to discuss over deeply rather than the earlier hospital pharmacy. Whatever the correlation with that particular drugs, earlier units, we have to discuss fastly rather than continuously. Yeah. Okay, today's session up to the unit 1 is over, and might be there is a seasonal exam announced by the exam department for 5th of October.